Hello students. In today's class, we are going to learn about prokaryotic translation process. Basically, what is translation? Translation is the process of formation of a protein chain by making use of a messenger RNA as a template or a source of information. So in this slide, we are going to discuss about what are the different components that are required for the translation process. So firstly, messenger RNA, which is basically the template for protein synthesis process, and it is read in 5' to 3' direction by the protein synthesizing machinery. Next are transfer RNA molecules. These basically act as adapters or carriers for amino acids during the protein synthesis process. The next component is ribosomes, which are the actual protein synthesizing machinery. Coming to enzymes, two types of enzymes are most important for the protein synthesis process. Firstly, the one that is required for bonding the amino acids to their transfer RNA molecules, known as amino acid transfer RNA synthetase. And the second enzyme is required for peptide bond formation between individual amino acids, leading to formation of a peptide or a protein chain. Apart from this, several soluble factors which are required during initiation, elongation, and termination stage of translation are required during protein synthesis. So here is a structure of transfer RNA molecule that act as adapters during protein synthesis. So it has a clover leaf-like structure. Important structural aspects to note over here is presence of this arm, which is known as the anticodon arm of transfer RNA. And the anticodon arm has a sequence that is complementary to the codons which are present on the messenger RNA. So this allows specific base pairing between the codons of the messenger RNA and the anticodons of the transfer RNA molecule. To the 3' hydroxyl end of the transfer RNA molecules are attached the amino acid during the bond formation between amino acids and the transfer RNA. Next component are ribosomes. So ribosomes are basically the protein synthesizing machinery, and they are made up of ribosomal RNAs. They basically recognizes the signal sequence which allow the initiation of the protein synthesis process. These also ensure accuracy of protein synthesis process by stabilizing the transfer RNA and messenger RNA interaction. They are involved in enzymatically linking amino acids in the polypeptide chain. They move along the messenger RNA molecule and read the messenger RNA in 5' to 3' direction. So here is the structure of typical prokaryotic 70S ribosome, which is made up of the smaller 30S subunit and the larger 50S subunit. 30S subunit is made up of 16S rRNA and 21 different proteins. And the 50S is made up of 23S rRNA, 5S rRNA, and 34 different proteins. Coming to the enzymes that are involved in protein synthesis process, amino acid transfer RNA synthetase, it catalyzes attachment of the transfer RNA molecule to its corresponding amino acid. Since there are 20 different amino acids, so there are 20 different amino acid transfer RNA synthetases, each enzyme specific for one amino acid. The attachment of the amino acid charges the transfer RNA molecule. The second enzyme required is peptidyl transferase, which catalyzes peptide bond formation between the amino acids linked by their transfer RNAs to the ribosomal P side and A side. They allow sequential transfer of amino acids to the growing polypeptide chain. Thus, during the end of protein synthesis process, we get formation of a peptide chain. The peptide chain contains an amino terminal end and a carboxy terminal end. The individual amino acids of the peptide chain are bonded to each other by a peptide bond, which is represented here, the CONH bond or the peptide bond.